It's time for another episode of Q&A in Science. I'm your host, Currio, and today I'm joined by a special guest, Kailee. Hi, Kailee. How are you today?
Hi, Currio. I'm doing great, and thanks for having me today. By the way, have you heard? Just last year, the first CRISPR gene therapy has been approved to be used on sickle cell patients. What a breakthrough.
Wait, tell me more. I know that these patients have red blood cells that are shaped like a sickle, meaning that they have lower oxygen carrying capacity and that they stick together and cause blockages in blood vessels. But what do you mean by doing therapy to a gene?
Have you heard of CRISPR-Cas9?
You mean the technique that allows you to edit DNA?
Exactly. Well, the basic idea of this technique is that there is some component, which is an enzyme called Cas9, that can recognize a specific sequence along your DNA and cut your DNA open at that site.
I always thought CRISPR works like a pair of DNA scissors. You mean to tell me it can only cut at specific places?
Yep. The system was first found in bacteria. Basically, it's their defense mechanism against invaders like viruses whose genetic material contains a lot of these specific sequences. Of course, it's not just a pair of scissors that can cut anywhere at once. You have to guide it to the correct site that you want to edit.
Doesn't that mean you must know the sequence of the DNA you want to edit before you can do CRISPR on it?
You're learning quick, Currio. Thanks to whole genome sequencing, scientists now know the sequence of many organisms, and there are software applications out there that tell you where in the DNA we can find these specific recognition sites. For example, Cas9 recognizes sequences where there are 2 consecutive guanine bases. These databases can highlight all the guanine locations within the gene of your organism. From there, we can design a short guide that directs the Cas9 to the precise spot we want to cut.
Oh no, but now what happens after your DNA is cut?
So the DNA has a mechanism that allows it to repair itself naturally called non-homologous end joining. If we cut it open at 2 spots, the DNA could repair itself without including that segment. This is how you can pick a specific gene and make it dysfunctional.
That sounds scary.
Alternatively, if you want to just change out a few bits in the DNA sequence or to edit a mutation, you can supplement the cell with the correct sequence so that the DNA will copy it and repair it via homology-directed repair.
I can see how powerful that is. So how is this used on curing sickle cell patients?
This technology doesn't actually directly lead to the production of normal blood cells. Sickle cell patients usually have a mutation in a gene that causes bad hemoglobin. When there is not enough normal hemoglobin, red blood cell sickle. The use of CRISPR here corrects the hemoglobin gene in blood stem cells, which are then transplanted back to the patient's bone marrow. This supplements the body with more normal hemoglobin, thus lowering the chances of red blood cells becoming sickle-shaped.
I see. So eventually, normal red blood cells will dilute the number of sickle red blood cells in the body. Why aren't CRISPR gene therapies approved earlier if they are so powerful?
The problem is that the effects of editing one's genes is still under debate. Some say it's like playing God. There's also a difference in editing your own body's cells versus cells in your germline, which are used in reproduction. Once you edit your germline, the effects will be passed down to future generations, which could have diverse and uncontrollable outcomes.
I can imagine enabling designer babies will blur the lines between nature and artificial creation. But to use this CRISPR technology on diseases is a good thing, is it not?
Actually, a lot of diseases are currently technically curable. For example, leukemia can be cured using CAR T therapy, which makes use of CRISPR technology. However, in Hong Kong, there is only one registered CAR T cell therapy drug, and the requirements for patients are very steep, so not a lot of people manage to get to that stage where they can get this treatment. This is why science education, policy, and legislation are extremely important.
With more people knowing the importance of science education and more young students being interested in joining research, hopefully this situation can change.
Whoa, Currio, that's probably the most insightful thing I've ever heard you say.
Of course, as soon as I find out whether I have the lazy gene, I'm gonna get someone to edit it as a medical treatment. Hey, since you are the expert, can you help me edit my genes?
Okay, slow down there. Just when I thought you were getting wiser. But yeah, you probably do have that gene. I'm sorry.
If I catch you, you're turning into crisps.
I'm kidding. That was good though.
Fine. I think it's also about time that we say goodbye. Thank you, Kailee, for your sharing today, and thank you everyone for watching. See you in our next episode, where we can continue to explore and discover together. Bye bye!